I will now ask Randi to join us. Randi will be presenting on how data are retrieved, entered, and organized. Thank you very much, Randi.
Okay, can you hear me?
Perfectly, thanks, Randi.
Okay, good. So I'm Randi Vita. I'm going to talk about how the data is retrieved, entered, and how it's organized in the IEDB. And I want to make a disclaimer that today is trash day at my house. So if a trash truck comes, I'm just going to mute and then unmute once it passes. So nothing will be wrong with your Zoom. It'll be on my end. But hopefully it doesn't happen. One already did pass, so maybe we got lucky. As Alex mentioned, the scope of the IEDB is infectious disease, allergy, automating, and transplant. We do have some HIV and cancer data, but that is only when there is 3D structured data for those HIV or cancer epitopes, showing a 3D structure of an antibody, a TCR, MHC molecule, or when we have antibody or TCR sequences for HIV or cancer epitopes, or if those epitopes appear in a paper that is within the scope of the IEDB. And all the data in the IEDB is from the published literature or some direct submissions. It's all experimental data. We don't enter predictions or review papers. So it must have been experimentally confirmed, showing an adaptive immune receptor binding to an epitope. As Alex mentioned, we do include negative data and lots of supplemental data. So each assay in the IEDB demonstrates the binding of an adaptive immune receptor, being a T cell or B cell, or MHC binding, or MHC ligand elution, where an epitope was naturally processed and presented by an antigen presenting cell. But once we know that that receptor is epitope specific, we capture all of its experiments. So if you have an epitope specific monoclonal antibody, we will capture that it also neutralizes whole virus, or that it also binds to a protein, or if a T cell line reacts to cross-reactive epitopes from another species, we will capture all experiments in that paper for any epitope-specific receptor. And in order to include data in the IEDB, we had to come up with some minimum criteria. And many people define epitopes differently. Often they're very small, but we wanted to capture as much data as we could and use this criteria across all papers and across all of our different curators going forward, so that all of the data was captured the same way. So we decided on any linear peptide less than 50 amino acids. If it was tested as an immunogen or antigen, we would capture that data. An author may go on to map the epitope more finely. They may then test overlapping 20 mers or overlapping 10 mers, or another author in a future paper may determine exactly the epitope. But we treat each paper individually and apply the same criteria. So we would capture all the data, whether it's peptides or small peptides or exact discontinuous residues that were shown to be important in recognition. So we have discontinuous epitopes also. And then we have non-peptidic epitopes. So these would be carbohydrates, lipids, often drugs, chemicals that cause an allergic reaction. And for them, they just need to be less than 5,000 Daltons to be included. And then for any experimental data to go in the IEDB, we have some minimum information that we need to know exactly the sequence for these linear peptides. If the author just tells us a protein name and its positions, that's not sufficient because there are many different isoforms for the same protein. Think about influenza, those proteins are highly variable. So at a particular position, you may have difference in sequence. So we always confirm the sequence. And then we also must know the outcome of the experiment. If we can't tell if IL-2 was produced or not, or if we can't tell if the antibody bound or not, we will need to clarify that with the author. And then the host whose immune response is being measured, was the antibody raised in a mouse or a human, for example. Surprisingly, that's not always present in a paper. So we will write to authors for all of these things because our goal is to put in as much data as we can. And so if any of this information isn't clear, we write to authors and hope that they respond. So you saw some of this curation process in Alex's slides. That we go to PubMed every 2 weeks, and we've retrieved more than 240,000 papers from PubMed so far. We capture all the data from the very beginning of PubMed until 2 weeks ago. And we also query the PDB looking for epitope relevant structures where a adaptive immune receptor is bound to its epitope. And then it runs through the automated classifier, as Alex mentioned, and that categorizes the papers and also looks for whether or not they would be curatable. And then there's a manual scan. And after this process, we end up with 44,000 papers that are likely to be within the scope of the IEDB and likely to be curatable. And then those go to the curators. And then the curators read the entire paper, and they find that sometimes it's a review paper, or sometimes we don't have enough detail and the author doesn't respond. So we ended up curating more than 21,000 references so far. And there's a full team of people adding to this data every week. And in the end, these are the categories of how the papers fall, being primarily infectious disease, then autoimmunity, allergy, and transplant. But this is entirely reflective of the literature. If suddenly there were a lot more epitopes published in autoimmunity, those numbers would go up. We don't bias it, we just go to PubMed and take what's there. So we have the data that we could find in PubMed. And if you think we're missing something, always let us know. So now Nina is going to show you a short video that overviews the actual curation process itself, how the curation team enters the data.
All right, here we go. Thank you.
Thank you for your interest in the IEDB. My name is Nima Salimi, and I'm part of the IEDB team. In this short video, my colleagues and I will give you a behind-the-scenes look at the process of populating the IEDB with data. Peer-reviewed publications are one of the primary sources of the data in the IEDB. A team of scientists called curators read and systematically extract the relevant information from these publications. This process is called curation. But before the curation process begins, the papers that are potentially relevant to the current IEDB scope, including infectious diseases, autoimmunity, and allergy, need to be identified from over 27 million publications cataloged in PubMed. This task is led by IEDB document specialist Lindy Edwards. Lindy, can you please provide us a brief overview of this process?
Sure. We've created a customized PubMed query that is run on a bi-weekly basis. The retrieved records are analyzed by an automated text classifier to identify any records that are unlikely to contain information that is relevant to the IEDB, known as uncuratable. The potentially curatable papers are sorted into broad categories and subcategories. For example, allergen pollen or infectious disease poxviruses. At this point, the potentially curatable abstracts are manually scanned by a senior immunologist on the team to confirm the accuracy of the classifier. We also rely on input from our users in order to make sure that we haven't missed anything.
Thanks, Lindy. In the next step, this list of papers enters the curation pipeline, where the curators will read and extract the data. The papers are assigned to curators based on the nature of the data described in the paper. For example, we have curators who specialize in curating papers describing non-peptidic epitopes, 3D structures, and papers reporting T-cell or B-cell receptor information. To capture the relevant data from the papers, each curator uses a web-based data entry portal consisting of both free text and controlled value data fields that help ensure data consistency. Debbie Shackelford, one of our IEDB senior curators, will now take us on a brief tour of the curation process using the curation system.
Thank you, Nima. The curation aims to objectively represent all the data that meet minimal IEDB inclusion criteria. To demonstrate the types of information extracted for the IEDB, the following paper is used as an example. In it, the authors use a conserved epitope found in the membrane proximal stem of influenza virus hemagglutinin and express it in a virus-like particle and then determine if it's sufficiently immunogenic to induce broadly neutralizing antibodies. I've highlighted two key words that are in the PubMed query Lindy just described that was used to identify the paper. The data entry process begins by adding a single epitope. The epitope structure, whether peptidic or non-peptidic, must be clearly provided in the manuscript or in a reference. Figure 1 in the paper provides a sequence of the 20 amino acid peptide found in the hemagglutinin of the 1918 flu. The epitope sequence is entered into the epitope field of the IEDB curation application. Each naturally occurring epitope is then assigned a source. The edit link accesses the molecule finder, which is used to find sources with 100% sequence match to the epitope. The molecule finder contains a subset of the entries you would find in GenBank and is continually amended as new sources are needed. In this case, multiple GenBank accessions contain the epitope sequence and the most appropriate is chosen. Once an epitope has been added, details summarizing the assays in which the epitope reactivity are tested are captured. In this case, a B-cell assay is used to capture antibody reactivity by ELISA. The assay details fields follow the temporal flow of the experiment, beginning with the host and immunization details, including the immunogen, the dose, and additional details, followed by the type of assay utilized, the assayed antibody, and the antigen used to elicit the response in the assay. There are custom sets of fields for B-cell, T-cell, MHC binding, and ligand elution assays. Additional details, such as MHC restriction and antibody isotype and sequencing, are also included when available in the manuscript. Automated validation based on relationships between data fields is extensively incorporated throughout the system in order to alert the curator of errors in real time. But once all of the information has been curated, the data can be advanced to our peer review stage.
Thanks, Debbie. As Debbie mentioned, an important final step in the curation workflow is the internal peer review process. Randi Vita, the IEDB Quality Control Manager, will provide an overview of this process.
Once a curator has entered all of the data for a given publication, the record is sent to peer review, where a senior curator rereads the same paper again and reviews how that data was entered into the IEDB's curation interface. We do this to ensure consistency across all of the different publications and among all of our different curators, following established IEDB curation guidelines. In addition to manual review, we also have interfield validation that's built into the curation interface itself. This validation checks for things like consistency among fields. For example, if the curator accidentally entered a greater number of subjects responding than were tested, you would get a validation error that looks like this. Both the curator and the reviewer can see these validation errors, and they must be resolved in order to promote any reference to the external website. Once the curator and the reviewer agree on how the data was entered and all of the validation errors are resolved, the data is then made public.
Upon passing these quality assurance and quality check steps, the data are made publicly visible in the IEDB website. The homepage provides summary metrics detailing the type and amount of data available. We always welcome your questions at help@iedb.org. Thanks again for your interest in the IEDB.
Okay, it looks like I'm not muted and my screen is being shown again. So I'll pick back up where I left off. So Nima just told you about the curation process and mentioned our consistency and quality control measures, which are really important to us because we want to treat every paper the same, regardless of what journal, what authors, what subject matter, and which curator enters the data. So we start with having a team of PhD level scientists who have specific expertise. They're all immunologists. They've all done their PhD, worked as postdocs, and had careers in immunology. So they do understand the papers that they're reading, and they know how to follow our curation guidelines. So we have formal guidelines that are a living wiki that's available on the internet, and anyone can access it. There's links to it from the IEDB website, and here is a link to that curation wiki. And then we have that peer review process that Nima just illustrated. And then we also rely heavily on immunological experts with the original design of the database, what the fields were going to be, what our scope was going to be, all of these rules we've run by external experts, not just ourselves. And then when we start curating a new subject matter or we encounter something new in the literature, we want to add new fields to the IEDB, we go again and again back to different immunological experts depending on the subject. So when the subject was non-peptidic epitopes, we reached out to non-peptidic immunologists. For cancer, we would reach out to cancer immunologists. We would always look outside of ourselves because we want to meet your needs as best as we can. And then also, as mentioned in that video, we have this validation within the curation application that helps the curators be more accurate, where they pick from existing terminology rather than typing. And then we have interfield validation, and we have validation that runs across the entire curation once they're done. And then we also get submissions. So the submissions will primarily come from epitope discovery contact holders, and they deposit into the IEDB directly. And as of 2020 of this year, we have 306 submissions. They're all that are publicly available, and we have 96 that are in process. And the amount of data that comes from these submissions is great because usually these epitope discovery contact holders generate very large data sets. So it amounts to 20 of the epitopes in the IEDB. And if you're interested in making a submission to the IEDB, contact Nina or support after the workshop. And the submission process is quite similar to the curation process. The submitters submit their files via a web application, just as the curators do. And that web application runs the same automated validation across their data as is what's run across the manually curated data. And additionally, we have a curator review what the submitter submitted and look that it makes common sense. And then they will work with that submitter if anything looks odd and make sure that it's accurate. And then we release this to the public when the submitter asks us to. Oftentimes they ask us to hold it until their publication comes out and they release it simultaneously. Sometimes it's they're waiting for a patent, for example. So the structure of the IEDB is that this is a database of experiments. To get into the IEDB, there must have been an experiment in that paper. So we start with the reference, as I mentioned, with that PubMed query. And for that reference to be relevant to the IEDB, it must have described at least one epitope. And each epitope must have been described in at least one experiment. Very often they're tested in many experiments, and we will capture them all. And for each experiment, we capture a great deal of detail. And I hope to illustrate why this is important to you through the query examples. So for each experiment, we have as many as 400 fields to capture all of that detail. And then we try and summarize that and package it to present it to you in a manageable format. So we describe the host whose immune response is being studied. What was the immunization procedure? Did they have an infection or were they injected with something? Or were they healthy and there was no immunization? The antibody, as Debbie in her example showed, we have capture information about the antibody isotype. Is it monoclonal? The effector cells, what tissue did they come from? Were they restimulated or not? Were they CD4s or CD8s or just PBMCs? And then every experiment has an antigen. And that's the antigen to which the immune response is being tested in that particular assay. So the same monoclonal may be tested for several different antigens, being the peptide, proteins, organisms, etc. And in order to enter all of this data in all of these fields, we like to collaborate with ontologies and external resources to make it simpler and to make our data more machine readable and interoperable. So ontologies provide standardized nomenclature for each type of concept, whether it's organisms or proteins or diseases. And that also gives us definitions, synonyms, and the hierarchical relationships between terms that these ontologies worked out for us. So by using those resources, we gain all of the information they already have. And this makes our curations easier. And also, I'm going to show you how we think it enhances your experience by using our finders. So I'm going to illustrate what these finders are. And then what they do basically is present the appropriate ontology to our curators and to you so that you can interface with the data that way. And this lets our curators pick from existing terminology and shows you all of the terms relevant to the data in the IEDB. And this helps us ensure consistency and accuracy, again, because our curators are not typing, let's say, for example, organisms. They're not typing out every organism strain that they see in a paper. They're choosing from that formal NCBI nomenclature. And this helps us find errors in our data also, because the relationships between terms in these ontologies allow us to enforce validation using those relationships and helps us find when there's errors of consistency. And then also, any other project that uses any of the same ontologies that we do is now interoperable with our data set. So the point of the database is to make everything easily searchable, but also interoperable, machine readable. So other projects using these same ontologies are storing now the same concepts and overlapping data sets using the exact same terminology and formal identifiers. So this is the first example that I think most people would be familiar with is the NCBI taxonomy. So this presents organisms in a tree structure. And so if the epitope is natural, it will be derived from an organism. And those organisms will be presented to our curators and to you via these finders that show the NCBI hierarchy. So there will be all viruses, there'll be all human herpes 5 viruses, and there will be specific strains of a virus. So the curator can pick the exact strain that is described in the paper, and you can decide to search for, if you would like all data for all viruses, you can do that, or all particular type of virus. I'll do an example of that query later. Or if you're interested in just human herpes 5 virus and you're not you don't care about the strain, you can search for that. And because we use the NCBI taxonomy and it comes with all of these synonyms, you can type much simpler things like C and V and still get human herpes 5 to come up because NCBI did that for us. And we organize all of the proteins, if the epitope is from a protein, using UniProt reference proteomes. So as the video showed for the curation, where Debbie was picking a specific protein that was 100% identical to the peptide sequence she was curating, that was a very specific hemagglutinin protein. But there are more than 400 different hemagglutinin proteins used to depict the IEDB data because all of those different isoforms have the different mutations that are important to the immune response. So we need to use them, but we want you to be able to navigate through this data in a reasonable fashion. So by organizing the proteins using the UniProt reference proteome, we present to you the set of proteins for each species. So organized using that same NCBI taxonomic tree, you can see all influenza virus proteins, influenza A virus proteins, and then we present to you the reference proteome package from UniProt, which comes with synonyms and the relationships between terms. And it also comes with these processed protein fragments. So UniProt further breaks proteins down into, here for this example, HA1 and HA2 chains. So UniProt does this for us because they're the protein experts and they've worked all this out. So our curators pick the right protein and then we run all of the proteins for which we have data through the UniProt reference proteome and make for you a nice pretty tree that's easy to navigate and has additional features and provides all the synonyms so that you can type more easily. And then some epitopes are non-peptidic, and non-peptidic epitopes are very complicated, especially if your field is not chemistry. As you can see, these carbohydrate names. So for those, we rely on the expertise of the CHEBI ontology. This is the chemical entities of biological interest ontology, and they're chemists, so they're able to create these structures that match what was in the paper that we were curating, so that you can get an accurate epitope and you can also get an accurate source of an epitope. And by using an ontology, we get definitions, hierarchical relationships, synonyms. There are often very many synonyms for non-peptidic structures. And I'll show you later when we look at the tree on how we present this to you. And for the host of the assay, who's immune response, we again use NCBI taxonomy to describe all of these different species for which we have data. And for diseases, we use the disease ontology. And this provides us with synonyms, definitions, cross-references to other projects, and relationships and organization of the different disease types. For example, all food allergies can be searched together as a set, even though they're curated separately. And then for the assays in the IEDB, we use the ontology for biomedical investigations, OB, and they organize all of the assays for which we have data in the IEDB. And this makes it easier on the curators in that they pick from all of the assay types, finding exactly what was mentioned in their paper. But we think that once we present this data to you using this finder that depicts the OB ontology hierarchy, you can see that you can search for all cytokine assays or all IL-2 assays, regardless of the platform used, or all biological activity assays, to whichever level is most relevant to you, because we don't know what sort of information all of our different users are interested in. So we present all of it this way, and then you can navigate the tree and search at what matters to you. And for the MHC restriction in the T-cell assays and also MHC binding assays and ligand elution assays, we present that to you using the MHC restriction ontology. And this ontology gives us the proper nomenclature for all these different MHC molecules. We have human, but we have many other species as well. And the MHC restriction ontology uses information from IPD and IMGT to present you with the proper nomenclature for MHC molecules, which can be quite complicated and actually does change over time as those fields evolve. So our curators and you, when you're searching, you don't have to type out yourself terminology with dashes, asterisks, colons, and slashes and get all of that right exactly. Instead, by using our tree structure, you can type a little bit and see what we have, or you can navigate through the tree, as I'll show you when I do queries, to see what sorts of MHC molecules we have data for and which groups you would like to search on. And by using the ontology, the data itself is modeled so that each complex is made up of two separate chains. So you can search on a chain whether it's part of a complex or tested individually. And we also have those organized based on the genetic locus that each chain was encoded by. And so the goal of the IEDB is to aggregate all of this data together from all of the papers, every epitope described in the paper, in every experiment that was performed. And one epitope may be tested in many different papers, or it may be tested only once ever in one experiment or many, many experiments. That's highly reflective of the literature and what people are doing out there. And so what's very important to you as users, and for me to explain to you during the queries, is that the same epitope may be positive in one context and negative in another. So whether or not it's an epitope depends on what your interests are. So this particular epitope may have been positive in an ELISA where the subject had a disease, and it may have been negative in an ELISA where the subject was injected with a protein. Or one ELISA may have been measuring the response to the epitope itself, the antigen, or the other ELISA may have been the whole organism and it was not recognized. So you have to look at the details in all of the data to determine what matters to you and what you think is an epitope that's relevant to your research. So that's why we're going to spend some time doing these queries to show you how to get at what matters and is most interesting to you. You know, the species of the host, the exact response type, the antigen, all of that determines what is an epitope. And then here again is the immunome browser, as Alex showed, plotting along the length of a protein from amino acid 1 to the end. That by aggregating all of that data from all of the different references and all of the epitopes that were curated, all together, when you perform your query, and this is the query I performed up here, epitopes from Timothy grass, from humans who had an allergic disease, that changes what the look of the graph is and where the epitopes fall. So this region, which is red, had negative assays, and the region in blue had positive assays in the context of my query. If I changed the host to not be human, the graph would change. Or if I changed that they didn't have allergic disease, the graph would change. So what is an epitope is entirely dependent on what your query is. And that depends on your field of research and that is what I'm hoping the second part of my talk today will help you get at. So this is the homepage, as you're going to be seeing a lot today. And you previously saw it, so I'm going to walk through it quickly. As mentioned before, you can learn more about us, see what we're up to. We used to have a lot of in-person conferences, which for now are all going to be virtual. And then the accounts of where we're at, which changes every week. The homepage is where I'm going to be spending most of my time today. I'm walking you through how to use it and what is hidden within all of these search panes. And as always, the question marks will give you more information on that concept. And then the tools, which we talked about a little bit today, but mostly tomorrow. And at the bottom of every page of the website is how you can get help and give us feedback, and we do want to hear from you. And then there's some hidden information up in the top part of the every page. There's an embedded search drop down called specialized searches, and within it there's an identifier search. And this is something that's often asked about, so I want to make a point to point it out to you. That you're interested in searching by PubMed ID, you can do that there on the homepage. If you have a specific epitope that you want to see if we added any more data to it and you have its epitope ID, you can go directly to that there, and all new data for that same epitope will be always added to that same identifier. And then next in that same blue bar header, there's another drop down for the tools resource. And then even above that, there's two sort of hidden drop downs for help. And within help, one of the users had asked if we had video tutorials, so I wanted to point that out specifically. Up in that far upper right-hand side, that help menu takes you to video tutorials. And this always is growing, so this workshop will show up here. If we have any other video tutorials, they'll be added to it, and older ones will be pushed down. But we do have quite a few tutorials that meet the criteria of what that user had asked about. And then under more IEDB, there's even more information. We've packed the website full of information, but I want to point out one thing here, which is the export. So export is also something people have asked about. You can download the entire database. This is a free resource. We exist solely to help you do your research. So feel free to take the whole database if you're a bioinformatician, and you can use it however you would like. From here, our CSV exports are actually the most popular ones, and they're here. This is further down the same page. It was a very long page, so I cut it in half. And then I want to point out two of our newer exports because they also answer some user questions. We have a full receptor export, and this will give you all antibody and TCR sequences in the IEDB. And for all antibody TCR sequences, we capture as much details as the authors provide. So if the authors gave us the nucleotide or full-length protein, CDR1, 2, or 3 sequences, they will be included in this export, and I'll also show you how to query on them. We also do gene usage if it's provided, and in this export, you will get each epitope that each receptor was shown to recognize. And then additionally, there's a 3D export that has all of the 3D structures in the IEDB, which is antibody, MHC, and T cell that are on separate spreadsheets. And this gives you a lot of detail about the PDB-IDs, the contacts, the IDs, the antibody, the chains, and you can combine this information with that receptor export for even more information. And so after the break, we're going to show you how you can query the IEDB, and I'm going to use your submitted questions to work through those queries. But now I believe we're taking a question and answer.
Excellent. Thank you so much, Randi, for that. I would like to invite Alex back. Thank you, Alex. I can see we've got a few questions coming through. So Alex, maybe if you'd like to take the first one while Randi takes a look. And
yeah, so there are the first two questions in the chat. One, how to use population coverage server for analysis. But it's a very good question, and we will talk about the population coverage tool in the analysis resource section later on today. Then there was another question about whether the IEDB provides bulk downloads, which I believe Randi answered with her slide just a minute ago. Finally, there is a question about standardizing HLA nomenclature. I don't know if Randi had a chance to look at it. Did you look at that question, Randi?
Yes.
So maybe you can take a crack at answering that.
Okay, so the person asking the question, asks basically the nomenclature is not standardized into a single format, and that an epitope might be HLA A0201, or it might just say A2 or A01 or something like that. And that's something we definitely deal with in the literature, but we've standardized it in the IEDB, in that we always present for you the precise nomenclature based on IMGT standards using that MHC restriction ontology. So you will always say HLA A0201 with the proper asterisks and colons in the right place because we use that ontology. Sometimes authors do only tell you that they have a class 1 restriction. And so sometimes you'll see vague information that way, but we'll never present the nomenclature incorrectly. But I'm not totally sure if that user was talking about tools or about the data. Within the data, it's always the proper nomenclature. And then I'll show a little bit more when I do the queries. So you'll see the full tree.
Yes, so I'm adding a little bit to what Randi was saying. So yes, it has been a continuous process in which we have been developing ontologies and hierarchies of HLA nomenclature. So in that context, it's always helpful to bear in mind that, for example, the user was mentioning, well, is it A02 semicolon 01 versus some references may just say A2.1 or A2. Some of the old data was generated with cytotoxic T-cell lines where they really didn't appreciate the fact that A2 had many different subtypes, so to speak. In that context, obviously, we can only curate what the author knew at that point in time. There is another question about whether the export that you can do can export both positive and negative data. Yes, that depends on what query you perform. But if you, in your query, you selected to only query for positive results, then you'll get only positive results. If you did not specify positive versus negative, you should be able to export both. I think that we do not have any open questions. So think you earned your break.
I think that's right, Alex. Thank you. There were only a few questions. Hopefully we can keep them coming in the other question sections that we have coming up. So yes, we do have a break now. It is about a 15-minute break, but we are running ahead of schedule. So let's reconvene in 20 minutes. So that will be 9:25 for those of you on the Pacific, but I'll pop it in the chat as well. So reconvene in 20 minutes. Go ahead, stretch your legs, maybe have some breakfast, lunch. And yes, we'll be back. I will pop in the chat when the team will be back. Thank you very much.